Hello, hi everyone. My name is Dr. Sana Khan, and today we are going to talk on the topic plasmid from the chapter Biotechnology. Plasmid is one of the very hit topic for your board exam as well as NEET exam. So let's get started and talk about plasmid in detail. So where do you find plasmid? Yes, plasmids are occasionally found in bacteria and yeast. So where are they found? They are found in bacteria and yeast. And they are rarely found. Okay, they are rarely found in plants and animals. So they are very often found in bacteria and yeast, and they are rarely found in plants and animals. Are you clear? So now you have understood where you can find the plasmids. So plasmid is very commonly found where? In the bacteria. So let us see where exactly in the bacteria you can see the plasmid. So this is a bacteria. What is this? Can you recognize this? This is the bacterial chromosomal DNA, right? So it's a bacterial chromosomal DNA, and this is the plasmid. So its plasmid is not the chromosomal DNA of the bacteria. Please do understand that. I think you must have already studied this in your 11th standard, right? That bacteria has a chromosomal DNA, and apart from that, it has an extra chromosomal DNA, which is called as plasmid, which is circular in nature. So it is small, double-stranded, extra chromosomal, circular, and it is autonomously replicating. Because of this property of plasmid, plasmid is very commonly used in recombinant DNA technology because it has an ability to replicate. And there are no vital genes which can be incorporated, and it hampers the desired product. Okay, so these are the few things about plasmid which you should know, and it doesn't need anything for survival. Okay, so the number of plasmids found in a bacterial cell is around 10 to 20 molecules. So this is a plasmid. What is the size of the plasmid? It is 1 into 10 raised to 6 by 200 into 10 raised to 6 Daltons, and it's a transferable genetic element, and the cloning size is around 0.1 to 0.1 to 10 kb. So here they have a thing called as replicons. Why they are called as replicons? Because they are capable of autonomous replication in a suitable host. Okay, so that is why you use plasmid, because they are able to replicate inside the host cell without any dependency. So they may get accumulated in bacterial cells in large number, like 100 per bacterium. So let us see few more characteristics of plasmid. So it's a minimum amount of DNA. Uh, it has a minimum amount of DNA. There are two suitable markers for identification, right? I have talked about the properties of a vector, in which I have already talked about the two suitable markers. Single restriction site. There are more restriction enzymes. Size range to 1 kilogram to 200. It can be cut with the help of restriction endonuclease enzyme, and it's a relaxed replication control, that is, it makes many copies. Is it clear? So these are the few characteristics about plasmid itself understood. So let us see few examples of plasmid which can be asked in your exams. So few examples are PBr 322, PBr 327, PBr 325. These are the few examples of plasmid vectors. See, I have already talked about who has introduced the plasmid, from which bacterium it was first tried to isolate it. You remember that I talked about Salmonella typhimurium, right? I have talked about Herbert Boyer. So we have already talked about this in the video called as cloning vector. So please, if you have not seen that video, go to the video of cloning vector and try to understand about vectors, and then see plasmid. You will get what am I saying. Okay, so if the genomic DNA get incorporated with the plasmid DNA, so it forms a structure called as episome. This is something which is not common, but it can happen that it may incorporate with the genomic DNA, and that time it is called as episome. Okay, so this is the plasmid DNA. Bacterial cell has its plasmid DNA. So the best advantage of plasmid DNA is, as it's small in size, their isolation and reinsertion in the bacterial cell is Very easy, because here what we have to do, we have to first isolate the plasmid, right, from the bacteria. After isolating the plasmid, we have to cut that plasmid. We have to make a space for our gene of interest, right, with the help of restriction endonuclease. Once that the gene of interest gets fit over here, okay, with the help of DNA ligase, it will be called as recombinant DNA. And then we have to insert this into the host cell, right? So we have to isolate as well as we have to reinsert for having a multiple copies. So this is the advantage because of its small size. Are you getting it? Okay. So let us see some nomenclature of plasmid. Very, very, very again important for your exam. PBr322. So what does this PBr322 stand for? Let us see. P stands for obviously plasmid. BR will stand for the name of discoverers, that is Bolivar and Rodriguez. 322 is a number designated. Okay, this is a number which is designated according to their discovery in that bacteria. So PBR322 is the most commonly used vector, and it is called as workhouse for gene cloning because it's the most common vector which can be used for biotechnology. Because it's very versatile, widely used, and easily manipulated vector. So which is this? Is this a natural vector or it is an artificial vector? Yes, it is an artificial constructed vector, because the vector which was normal, it not natural, it was found that it was not having all the characteristic for gene cloning. So the scientists, what did they do? They created their own plasmid, that is your PBR322. Obviously, the other plasmids were also created, but PBR322 was the most common one which was created and being used in biotechnology for gene cloning. Okay. Another example, let us see PUC 18, where P stands for plasmid. Your UC stands for the name of the place where it was discovered, that is University of California, and numbers designated is 80. Okay, so here the number designated is 80. So that's all about your plasmids. A small quick video, and in detail also there are certain things about plasmid which was already talked in the cloning vector video. That's why I didn't repeat it over here. Okay, so that's all about plasmid. Soon I'll meet you with the next video. Till then, bye, take care, study hard, and work for your dreams. Bye.